Hi, this video is about proteomics. We have seen genomics before. Genomics is the comprehensive study of genome. Just like that, proteomics is the comprehensive study of proteins. So, proteomics can be defined as the comprehensive study of all the proteins of a cell, tissue, body fluid, or organism. So, we can study the proteome at different levels. It aims to obtain more global and integrated view of biology by studying all the proteins of a cell rather than each one individually. So in proteomics, we are studying all proteins present in a cell together and how they are interacting each other, how they are affecting each other, or how they are conducting the signal transduction. So in the proteomics, we are studying the structure, function, expression profile, protein-protein interaction of the proteome. So it includes so many different levels of study and so many different types of study. And the peculiarity of the proteome is like it changes all the time. It's a dynamic nature. It has a dynamic nature. When compared to the genome, which is used to be stable, because all the cells in our body is having the same genome throughout our lifespan in most of the cases, and only there is only very rare exceptions. But proteomes are different. Proteomes will change time to time. The cell change the expression of different proteins time to time. It change the proteome change will cell type to cell type. If you are taking the neuronal cell type, the protein expressed by the neuronal cells will be different than that of the protein expressed by liver cells. But both cells, the genome will be the same. Okay, so the proteomes are more complex and more dynamic, and it also change in response to different signals, like the signal transduction, the endocrine signals or paracrine signals or autocrine signals. Every type of signals and stimuli can actually change the protein composition of the cell. So when compared to genomics, the proteomics is more complex, more dynamic, and it needs different types of techniques to study the protein dynamics. So the proteomics include the characterization of proteome, its expression profiling, structural analysis, functional analysis, and also studying the interactions and modifications of protein in different stages of development, different stages of response, how they are actually receiving the signal from the outside the cell and how they are responding to the different signals, how the homeostasis is maintained, how different metabolic pathways are controlled. Everything in a cell, all the functional aspects, almost all functional aspects of a cell is actually controlled by proteins. Of course, DNA is having the genetic information, but the functional unit, proteins act as more of a functional unit rather than the blueprint. And it has many applications in diagnosis of disease and monitoring of disease progress, especially in the case of cancer. The proteomics is a complex area of study, so it includes different sub areas, which includes protein expression profiling, how the proteins are expressed in different types of cells or different stages of development or in response to different stimuli, is actually studied in protein expression profiling, which is a part of proteomics. And to also have post-translational modifications. Proteins can be modified in different ways. Based on the modifications, its properties will change, so it's another area of study. Protein-protein interaction, how the different proteins are interacting each other for signal transduction, maybe for metabolism, maybe for making the multimers or the protein complexes or enzyme complexes. So that's another area of study. There is structural proteomics, then functional proteomics or proteome mining. So the proteomics has several subfields, and on each of these subfields is having different applications too, which is also indicated in the figure. Importance of proteomics. Proteomics is having very significance in research as well as in understanding the basic biology. So as I said before, proteins are the functional unit of the cell. They are the active component which is involved in the metabolism and also signal transduction, and how they are sensing or identifying external stimuli, maybe physical stimuli, chemical stimuli, or the ligand based stimuli, and how the cell is responding to that signal, how the structure is maintained in the cell. Everything is depending on the proteins, how the proteins are the functional units. So studying the proteomics has many significance, and it is very important for research as well as for basic biology. In the proteomics, it used to investigate many different questions: when and where the proteins are expressed, as we know, All proteins are not expressed in all type of cells. The proteins have selective expression and dynamic expression. So when and where it is expressed, the rate of if it is produced in a cell, what is the rate of production, degradation, and steady state abundance. Then how they are modified, especially the post-translational modifications, and the movement of proteins between subcellular compartments, like the proteins are synthesized in the ER and it is then moved to the Golgi, and it may be moved to lysosome or the extracellular space, how it is moving, what's the dynamics, and role of proteins in metabolic pathways and signal transduction, and how they are interacting each other. So proteomics is investigating different types of importance questions. Then significance of proteomics. Many type of information cannot be obtained from studying the genomics. The genomics, we will get the genomic data and how we can actually identify the open reading frames, and we can actually predict the proteins which are made from the genome, but it is incomplete because of the following reasons. First one is annotation of the genome. How do we find the function of a part of genome? So most of the functions of genome is actually based on the functions of the protein. So in order to annotate the genome, we should know the function of the protein. And the complication in knowing the function of a protein is like all the open reading frames in a genome do not code for a protein. Okay, if it may have all the components for protein expression, it will have all components for transcription or translation, everything, but it may not be expressed. We have several examples for that. So all the genes we are identifying using genomics or the bioinformatics tools do not actually code for gene or do not actually produce the gene, produce the protein. And the exon intron concept is even though we know the common sequences in an exon and intron, cells used to act differently. We cannot actually identify all exons and introns using bioinformatics. Then functions of a proteins cannot actually all fully understand by the bioinformatics tools alone. We need to do the physical experiments or the proteomics experiments to really understand the function of a protein. So basically, to annotate a gene or finding the functions of a gene or importance of a gene in a genome, we need to know the function of the proteins, and the function of the protein is actually studied in the proteomics. So that's an important significance of it. One importance of the proteomics. Then we have the protein expression studies. We can actually study the mRNA level studies, but not all mRNAs are converted into proteins. So there are different stages in this figure. You can see if the DNA, the genomics deals about DNA, but all the genes in the DNA is actually transcribed into RNA. And once it is in between the DNA and RNA, we have transcription, and it is controlled by transcription regulation. And when RNA is become mRNA or mature RNA, there is a possibility of alternative splicing. One mRNA can be spliced in different ways and making different proteins. So that's another level of complication. Then once the mRNA is made, there is translation regulation. Not all mRNAs are expressed in the same way in all type of cells. It will change. And once it is made the proteins, there are different mechanisms which actually control the protein, which include the proteolysis and post-translational modification and compartmentalization. So it's a very complicated scenario to actually understand the expression of a protein. So how much amount the proteins are expressed. Sometimes people used to quantify the mRNA, and they actually want to make a correlation between the mRNA levels and the protein levels, but that doesn't really work. The levels of mRNA in a particular type of cell do not actually correlate with the protein levels, because there is translation regulation and there is also post-translational regulations which actually control the protein levels. Then protein functions. To fully understand the protein function, we need to have the three-dimensional structure of the protein, especially if it is a novel protein. So proteomics actually helps to understand the function of protein. Then protein modification, post-translational modifications of proteins is very much important for regulation of the proteins. Not only enzymes, the signal transduction and metabolism, it's important. So protein modifications are studied in proteomics. Then protein localization and compartmentalization. Protein used to have different functions in different compartments. So where is it actually located? Whether it is located in the lysosome or it is in the mitochondria. So depending on the location of the proteins, its significance will change. So that's studied in proteomics. Then finally, the protein-protein interaction. How this protein is interacting with the other type of protein is also studied in proteomics. So that is the actual importance of proteomics over genomics, or how it is complementing, the proteomics is complementing genomics. Then proteomics has so many challenges when compared to genomics. There are different challenges, especially in eukaryotes. In eukaryotes, there are more proteins than genes. So because if we take the example of humans actually have about 30,000 genes, and from these 30,000 genes we are at least producing 85,000 different proteins. So if you look at the genome, we will see only 37,000 genes and hardly 30,000 genes. We identified actually 25,000 genes. If you are adding everything together, there will be actually 30,000 genes. These 30,000 genes are producing 85,000 proteins. So there is a huge difference. This difference is arising due to alternative splicing and post-transcriptional modification and also the post-translational modification. So these all contributed to increased number of proteins. Then the other thing is that all proteins are different. They have different properties. They have different physical properties, different chemical properties, structural properties, enzymatic properties. So they are a very diverse group. If you take the DNA, all the DNA is having same structure. Almost all DNA is having similar structure. So it is actually easy to identify the DNA or manipulate, do experiments with DNA. But when we are trying to do experiments with the proteins, all proteins are different. So one method suitable for one protein may not be applicable for other proteins. So the proteins is a diverse group and the quantity is different. So it is very difficult to do experiment with the proteins. So the proteomics is a tough task. Then type of proteomics. Depending on the use or the objective of the proteomics, it is actually divided into different groups, which include protein expression proteomics. As the name implies, it's actually quantify the expression levels of different proteins in different types of cells, or expression of protein in different conditions. Maybe if you take an example, how a cell is producing different proteins in a normal case or in a case of diabetic patient. So we can actually compare what make the diabetes, what are the changes caused by the diabetes, or we can compare cells which is growing in a normal temperature and cells growing in a higher temperature. How cell is responding to that situation, how the protein expression profile will change. So, or the structural proteomics. Second one is the structural proteomics. It's aiming to identify the structures of proteins and what are the structures of the protein complexes, and how to identify the presence of proteins in different organelles. All are done in structural proteomics. Finally, we have the functional proteomics. It's actually a broad term for many specific directed proteomics approaches, and it actually select a specific group of proteins and actually try to identify the functions, like the proteins identified in a signaling pathway or proteins identified in a metabolic pathway. So in the functional proteomics, we have more functional aspects, studying the more functional aspects of a protein, not a single protein, a group of proteins, how they are interacting each other, how they are linked in a metabolic pathway, or how they are linked in a signal transduction. And depending on the type or the approach, the proteomics can be a top-down proteomics or bottom-up proteomics. Here you can see top-down and bottom-up. In top-down proteomics, what we do, we actually isolate the protein, purify them, and we are doing the experiment in the purified proteins. So here you can see protein extraction. After these proteins are separated and they are purified, then we are doing the experiments in the purified proteins. And so before doing the experiments, we actually know which protein we are working on. Such type of proteomics is called a top-down proteomics. The bottom-up proteomics is also called a shotgun proteomics. It is just like the shotgun approach in the metagenomics. So in shotgun mechanism, what we do, we just extract the protein. So if you are extracting the protein from a tissue, we will get many different types of proteins. And what we do, we actually do not isolate the protein or purify the protein. Without isolation and purification, we actually convert it into peptides and do the analysis. So such type of proteomics is called bottom-up proteomics. So in top-down proteomics, we are purifying the protein and we know which protein we are working on. Okay, but in bottom-up approach, we do not know which protein we are working on. We are isolating a whole protein from the tissue, cell, or organelle, and we are digesting it, and we are conducting the study. After conducting the study, we are actually looking for the homologies, and we are trying to stitch in all the proteins to get the whole structure of the protein. So it's like a shotgun approach. And the proteomics have its various applications. I am not going to give a detailed description of the applications. So in brief, it has many applications in personal medicine, So we can actually personalize. Maybe in the case of tumor, it has many applications, because the studying the protein profile, protein expression profile of a cancer cell or the cancer tissue will give a better idea about progression of cancer, and what are the medicines can be used. So it can be used for personalized medicine, and it is extensively used for biomarker discovery for lifestyle diseases or cancer or any other diseases. So using this biomarker, we can actually identify the presence of disease very much ahead of time. Then it's used in drug discovery and drug development. Then system biology, agriculture, food science, pleioproteomics, and astrobiology, everything actually use this proteomics approaches. Thank you.